So a little bit about KRAS lung cancer, of which I think a lot of it is re-emphasizing things that Terry already talked about. KRAS mutated lung cancer is actually the most common oncogene-driven subtype of non-small cell lung cancer. So in the United States alone, over 50,000 individuals are affected every single year. And these are new people who are diagnosed. And it accounts for about a quarter of people who have non-small cell lung cancer, which is the most common subtype of lung cancer. And what I've illustrated on the right is, even though we classify this group all as KRAS-driven lung cancer, it actually has multiple subtypes that are dependent on the actual specific mutation. And so it's important not to know just that someone has KRAS lung cancer, but what specific mutation someone has. And that's the reason that's important is the medicines that are available for it are different. And so what I've tried to illustrate in these three pie graphs are the most common subtypes of KRAS lung cancer. And so the vast majority of individuals have KRAS G12 something, and there I've listed the kind of wedges of the pie of which ones are the most common. Other individuals have G13 or Q61, and those tend to be the most common types of KRAS-driven lung cancer. So next slide. So what exactly is KRAS? So illustrated here on the left-hand side are proteins that are outside and inside a human cell. And the more common subtype of lung cancer people may have heard about is something called EGFR lung cancer, which was discussed earlier. And that is what's labeled as RTK. So that's a protein that gets mutated on the surface of a cancer cell. And KRAS is actually a protein that's right after it. And this entire pathway of proteins all linked together tell a normal cell to divide and persist. So KRAS is actually a normal protein in all human cells. And once KRAS protein is turned on, so that's what I've illustrated here is the KRAS is in purple, and you can see that I put some arrows in. There's an off state of KRAS, which is KRAS, it's called KRAS GDP. So that's when the signaling is off. And then it can shift to KRAS turning on, which is KRAS GTP. And when KRAS protein is more in the on state, that's when it tells the rest of the cell to grow and persist. What I've illustrated on the right-hand side is an actual picture depicting KRAS protein. And the three colors indicate where the 12th amino acid is, the 13th, and the 61. And so this is a schematic just showing what areas of the protein itself can be mutated in KRAS lung cancer. And then when one of these sites is mutated, the entire pathway is on when it's not supposed to be on. So cells are now growing and dividing and persisting when they're not supposed to. And that's when a normal cell becomes a cancerous cell. So the next slide. So it turns out KRAS actually is one of the first cancer proteins that was discovered. So this was first identified back in the 1960s on the far left-hand side, where scientists realized that there was a virus in rats that led to sarcoma. And this led to the development of what's called the oncogene theory. So it's saying that a gene, just a single protein mutation, can turn a normal cell into a tumor cell. And then KRAS was one of the first oncogenes that was cloned. So we learned more about the sequence of KRAS. And it wasn't until 2013 that we actually were able to develop a drug that can actually turn off the mutant form of KRAS protein. And since then, there's been a acceleration of new discoveries in the field. With just last year, the US FDA approved the first KRAS-directed medicine against KRAS G12C, specifically in lung cancer. And since then, there have been multiple other medicines that are being tested in the clinic, not just for KRAS G12C, but other subtypes of KRAS cancers as well. So next slide. So to talk a little bit more about this discovery, here I've illustrated what the actual medicine does to the KRAS protein. So shown in the blue circle is the binding site of the GDP, which is a molecule that is bound to the on-off form of KRAS. And what happens is these scientists were able to find a separate pocket, which is illustrated in the yellow box, that had previously been unknown. And they were also able to develop a medicine called sotorasib that can actually covalently bind, so just lock the entire KRAS protein itself in this off state so that it's no longer turning on. And sotorasib specifically binds to the KRAS G12C mutant form of the protein, turning the entire protein off and preventing the cells from growing and dividing. And so this was a huge breakthrough in 2013 when we were finally able to develop medicines for KRAS. And since then, borrowing upon these principles, we are now developing new medicines for other KRAS drugs. And we're also, or other forms of KRAS lung cancer. And we're also developing not just these small molecules, but other new ways of binding to and shutting off KRAS. And so it's been very exciting to have this new discovery in 2013, and it's only accelerated research and discovery in the field.